I'm seeing you attending lecture from a study related to post-harvest handling of fresh commodities. Dr. Pankaj Patrare is teaching you some interesting aspects about the post-harvest handling practices. And as I aware, today morning session was related to the economics behind the post-harvest technology. So in this session, I would like to highlight the importance of post-harvest technology and the recent innovations which is taking place in the field of post-harvest and technology. If you see, cold plasma or treating the fruits and vegetables or extending the shelf life of fruits and vegetables using a cold plasma is a recent technique. In past decade, there are a lot of researchers and a lot of scientists working on this cold plasma technology and exploring its potential in extending the shelf life of fresh fruits and vegetables and exploring its application in extending the shelf life of fresh commodities throughout the market. So in this particular lecture, we'll see some introduction about cold plasma, and first we'll see what is plasma and what is the difference between the cold plasma and hot plasma, and what are the different systems available for generation of cold plasma, and mechanism of inactivation of microorganisms using a cold plasma as a tool, and possible application of cold plasma and plasma activated water in food preservation, application of cold plasma in shelf life extension of fresh commodities, and a conclusion. So as you all know, there exist four, we usually say there are different states of matter. So three states: solid, liquid, gas, which we all know. So solid is nothing when the atoms are tightly packed in a regular pattern. When the atoms are tightly packed together as a solid matrix, we call it as a solid matrix. And one electron or one energy state excited from solid state, we have liquid state, where the atoms are closely packed together but not in a fixed matrix. It can be randomly moving together, which is close together with no regular arrangement, vibrate, moves about a particular lattice and side past each other. So that is liquid phase, whereas gaseous phase is well separated with no regular arrangements. It vibrate and move freely at a high speed. So that is the difference between solid matrix, liquid and gaseous phase. Right. Now if you excite the electrons from gaseous phase, it becomes a fourth state of matter, that is plasma. Beyond gaseous phase, when you are increasing the energy level of a gaseous molecules, or when you are exciting the gaseous molecules from gaseous phase, it becomes another excited phase, that is plasma. So plasma is we can call it as a fourth state of matter. It has no defined volume or shape, and it composed of electrically charged particles. So if I say plasma, like it is a fourth state of matter, it's not that it doesn't exist in the nature. We have plasma existing in nature in different forms. If you see this interstellar space, where the stellar atmosphere or the gaseous planets, if you see the gaseous planets, gaseous nebula, these are made up of plasma only. And we can say this, our ionosphere, the Earth ionosphere is made up of gaseous plasma, mostly our ozone. And we have Van Allen radiation belt, which is also an example for a naturally existing plasma. And we have solar wind, that is also a form of plasma. And you can see the sun itself, the whole sun itself a ball of plasma made up of a nitrogen, mostly made up of a hydrogen gas. So it's a giant ball of plasma. If you see in Earth, you can see this excited state of gaseous phase when we are having a thunder and lightning. So it gives a excited state of plasma. That's why you can see glowing of this lightning. It is made up of plasma. This is also a plasma. Here, there you can see a high voltage high energy, where it's ionized the gaseous molecules around. So that's why we see this kind of lightning. And in the northern hemisphere, this the naturally occurring phenomena, aurora borealis, is also a kind of plasma. It is an ionized form of gas. So the gaseous molecules which is around is getting ionized, and the ionized state of gaseous molecules emits the green color light. Okay, that is the reason behind the aurora borealis in the northern hemisphere. And man-made plasma, we have plasma lights, right? So now we have LED lights, right? LED tube lights. Before that, we have fluorescent bulb, fluorescent tube lights. Fluorescent tube lights, and you have neon lights. Neon light, when you fill neon gas or argon gas or any other inert gas inside, it produces its characteristic color. So we have electrode in between either end of a tube light, and we pass electricity. When you subject the gaseous molecule in between to get ionized with the sufficient voltage, it get glow. So that is the reason the tube light emit lights. That's an inflorescent. That's a light which is emitted from plasma. So the fusion plasma, fluorescent tube lights. Fluorescent tube light is a best example for man-made plasma. It is inside the bulb. Now what we are going to do in case of our plasmides, we are making a plasma. The plasma should come in contact with the food material. So whatever we are going to deal with this plasma application is, we have to make a plasma, and somehow we have to make this plasma to come in contact with the food material for further process. It can be a modification process, or it can be a sterilization process, or it can be just a cleaning process. So there are a number of applications for cold plasma, right? So only thing is, we have to make a plasma outside, and the plasma has to come in contact with the food materials. Okay. Now let us come to the plasma production. Plasma production is nothing like we are going to do ionize the gas is small. The ionization of neutral ground state atom or molecules or radicals by an electron impact. By impacting electron with one another is an easy way to produce ionized gaseous molecules. So this is the most predominant ionization mechanisms which we use in most of the plasma production techniques. And if you see the chemical composition of the plasma, it has a mixture of gaseous molecules, free radicals, carbon dioxide or nitrogen gas mixture, or singlet oxygen, ozone, nitric oxide. There are different types of ionic particles, or you can see excited state of ionic components. This is the chemical composition of the plasma. And the primary factor which is responsible for the inactivation of microorganisms is the reactive species. The reactive species which is produced during this excited state of ionization is responsible for inactivation of microorganisms, especially this singlet oxygen, ozone are responsible for destruction of microorganisms. I will explain you the mechanism in the following slides. And plasma generation, the basic principle is we need a high voltage power supply. A high voltage power supply, or we can produce microwave plasma, or we can produce a magnetic field, electromagnetic field. We can aid the plasma production, and the radio frequency can also aid plasma production. So already we have an electromagnetic field where already the electrons are excited, which can be very easy to produce a further plasma. And we need a feed gas. As we are going to ionize the gaseous phase, we need a source of gas. If it is atmospheric gas, it has a mixture of gas composition, and then our resulted plasma will also be having a mixture of reactive species. Whereas if you want a pure source of a pure or a separate source of reactive species, oxygen reactive species, then we have to selectively give gas composition. If you use oxygen, it gives oxygen reactive species, it produces oxygen reactive species. Every reactive species has its own characters. Nitrogen reactive species are useful in some kind of operations. Oxygen reactive species can be used in some processes. As I said, there are a number of applications for plasma. Every one can be used in different applications. Generally, we use atmospheric gas or nitrogen source, nitrogen gas, or oxygen separately, or any inert gases also like argon, helium gas can also be used, or combination of gas can also be used for plasma production. So plasma, it has mixture of gaseous molecules, atoms and charged ions, free radicals, free electrons, ozone, elemental oxygen and photon. So these are the composition of plasma. And if you see the delivery system, it depends upon the method of plasma production we are adapting. So there is a corona discharge method. When you use a sharp edge to discharge the charged particles, then it is coronal discharge. Atmospheric plasma jets available, and then pulsed plasma, it produces plasma in pulses, and then microplasma, nanopulse plasma. So there are different types of plasma production techniques. So these are some of the most widely used technique. One is corona discharge and gliding arc discharge and dielectric barrier discharge, radio frequency plasma. The same, when you are subjecting this corona discharge inside a radio frequency cavity, we can make radio frequency plasma. So basically nothing, we need an electrode. We need a ground electrode and another electrode which is connected to a high voltage power supply. When you are allowing the ionization to take place in between these two electrodes, it produces plasma. Okay. So the technical parameters in a plasma system, the plasma parameters depends upon applied current, potential difference we are giving, gaseous composition we are using, either atmospheric gas or a separate gas, geometry of the electrode, the distance between the electrode or the type of electrode, whether we are going for a pin to plate, we have a pin plate, usually we use pin plate cold plasma discharge. One electrode will be a plate, another electrode will have number of pins. Pins means pin headed electrode. So this pin head will allow discharge of electron easier, and then the pressure inside the plasma production chamber. Even in atmospheric condition we can produce plasma, that is atmospheric pressure plasma, atmospheric pressure cold plasma, whatever we make in atmospheric pressure. Even at a higher pressure also we can make plasma. Even in vacuum pressure also we can create plasma. So that will become vacuum pressure plasma or high pressure plasma. It can be characterized because based on the pressure conditions which we are using to generate the plasma. And then the type of plasma generation, as I said, either corona discharge plasma or a parallel plate dielectric barrier discharge plasma. So there are different types of plasma based on the arrangement of electrode and the voltage applied. And if you see applicator, usually we generate a small portion of plasma that takes a lot of energy. If you want to make a large quantity of plasma, we need a higher voltage power supply, and we need a higher sophisticated equipments like a closed chamber, pressurized chamber, so pressure treatment. So these kind of applicators are needed for higher production of plasma. And then if you see number of the dosage required for a particular treatment of plasma, temperature, process of the temperature. Usually as it is a cold plasma, there is no temperature increase when it is exposed to plasma. When the food material is exposed to cold plasma, there is no temperature increase. it remains under the same atmospheric temperature conditions, but there will be a lot of changes. The changes is based on reactive species which is exposed to the food materials. Now, if you see the plasma, you can see the plasma will consist of most of the, as per my experience with plasma experiments, when we produce plasma, it has major composition of its plasma will be ozone and UV radiation, visible light, electromagnetic field, thermal radiation, electrons, single electron, and the excited species or reactive species which is produced from the gaseous molecules. So these are the major composition of the plasma, and when each and every portion of this plasma has a different types of reaction with the food materials. Okay, this excited species or the reactive species are highly reactive. It immediately react with try to react with some other molecules. When it try to immediately react with other molecules, either it makes another molecule excited or ionize another molecule, or it break down the molecules. It break down the molecular structure of a food materials. So that is what this excited species or reactive species do, and this electronic electrons as well. The singlet electrons or positive or negative electrons will also do. If you see the reactive oxygen species, example, if you are going for a plasma production with oxygen alone, it is associated with antimicrobial activity. Mostly when you produce plasma based on oxygen, it can be widely used as a disinfectant, or it can be used as an antimicrobial plasma, because it has ozone as a major component. Ozone as a major composition. It has hydrogen peroxide and ozone, hydroperoxyl, peroxyl and singlet oxygen, hydroxyl radicals, carbonate anion radicals. So these are some of the composition of the reactive species which can be used for inactivation of microorganism. So same we have this reactive nitrogen species, like we can nitric oxide, nitrogen dioxide, peroxynitrile, nitrous oxide, alkaline peroxide nitrate. So these are some of the reactive nitrogen species which is produced when you are making plasma from nitrogen gas. So if we come to the application in food industry, it has wide application. Plasma can be used as I said, it can be used for surface sterilization also. Degradation of pesticide, removal of pesticide from the surface of fruits and vegetables, removing the microorganisms from the surface of fruits and vegetables, and then even in case of meat and fish and meat product, it retains the freshness and it destroys surface microorganisms, and then in cereals, pulses, it is used to reduce toxins. If you see aflatoxin content in case of groundnut, aflatoxin content in case of chili can also be degraded. The aflatoxin content can be degraded using plasma, and it can be used to remove pesticides from the fruits and vegetable surface. So there are a number of applications which can be used in case of plasma and plasma activated water. So there are two kind of application. Plasma can also directly applied over the fruits and vegetables or any other materials to do some process in it to make some changes in the food product, or it is also used in extraction, like if you see ultrasound assisted extraction. So using ultrasound we break down the cellular structure with ultrasound. Similarly, this plasma has a etching property. When it is exposed to the surface of fruit, it causes cell wall degrade the cell wall component, and it allows enzymes to react, and it allows biotic components to come out. So it has some um, application in case of extractions as well. And if you see plasma, um, plasma activated water is, to make a plasma activated water, we need to expose this plasma to water, and the water will absorb, or water will dissolve this reactive species which is produced from the gaseous molecules. So the reactive species which is produced from plasma will be get dissolved in a water. So the reactive species can be again can be made reaction with another molecules when the plasma activated waters are used. So it can be used for bioactive components, functional property, physicochemical properties, and sensory attributes. So there are different applications, as I said, different. When it reacts with the phenolic component, it make oxidation of phenolic component, and it lower the intensity of plasma. Whereas in case of vitamin, when it reacts with vitamins, it is highly sensitive heat vitamin. It highly sensitive heat vitamins. Some of the heat vitamins, it retains the heat vitamins, whereas the lower treatment time and intensity. So it can destroy other microorganisms and other all those things. And you have antioxidant activity, enzyme oxidation, reduction of enzymatic activity, and modification of carbohydrate, lipid oxidation, enzymatic reaction, and protein denaturation. So these are some of the possible changes which can occur in case of food materials. Reduction in the moisture, reduction in the total soluble concentration. So these are some of the changes which occurs to the chemical and nutritional parameters of a food materials. So if you see, this is the plasma. If you see the plasma, the blue color plasma, as I said, it comprises of two regions. One is active region and the remote region. Active region has a whole lot of positive and negative ions, separated ions. When it is coming to the remote region, only this radicals, only these radicals will get exposed. This electron and the positive electron, the cluster, the ionic clusters are get degraded immediately within the active region itself. So the disadvantage of plasma is, when the plasma is produced, the half-life of component which is produced as a result of plasma generation is very less. The half-lifetime of these components are very less. It immediately degrade, or it immediately combine with other molecules to form other molecule. So as soon as possible, if it is possible, as soon as possible, when the plasma is produced, if it is possible to expose to some other material, it can be used for multiple application. As I said, protein and enzyme. When it is exposed to protein, it denatures the protein, or it breaks down the amino acid, oxidize the amino acid. Loss of enzymatic activity. It reduces enzymatic activity rapidly when it is exposed to enzymes. And we have lipid and fatty acids. The membrane of lipid peroxidase, it degrades the membrane of lipid peroxidase through production of a superoxide anion and hydrogen peroxide. And we have cell wall breakdown. Cell wall breakdown is one of the common character of a plasma when it is exposed to a fresh fruits or fresh food material. It may break down the cell wall, the breaking of chemical bond, or you can see etching or erosion of cell wall component by bombardment of radicals, free radicals over the surface. It increases in the surface roughness. It increases the surface roughness. So this is the one of the drawback in case of fresh fruit. The same drawback is advantage in case of seed treatment. If you see the seed treatment, germination of seed, to removal of seed coat, they can use this plasma. it itches the plasma seed coat surface, and it aids easy germination of seeds. In case in certain studies, they are using this plasma as a seed treatment also. And then cell wall membrane, it perform etching of cell wall by bombardment of free radicals, a diffusion of reactive species, and consequently it causes local damage to the cell structure. And then it causes damage to the DNA and RNA of a microorganisms, and it also causes reduction in the cell multiplication. Thereby, it also reduces number of log cycle in case of harmful microorganisms. So, the predominant reasons for inactivation of microorganism using a plasma is degraded lipid molecules, proteins, and DNA of the microorganisms. Lipid oxidation, especially on the cell membrane, is also one of the major cause for cell death, and protein denaturation of cell wall, oxidation of amino acid and nucleic acid, cell wall ruptured due to the electrostatic force. The electrostatic force created by bombardment of free radicals over the surface of the cell, and the deformation and leaking of bacterial chromosome or cell wall material. These are responsible for cell death or cell lysis when it is exposed to microplasma. This is the dielectric barrier discharge, a pin to plate plasma device. So you can see the plasma, how the plasma is generated here. We have to change this. So you can see we have a ground electrode plate, and there is a pin type electrode at the top electrode. In between you can see the plasma generation in between. So when this is very closed space, so you can see a plasma generated between these two electrodes. Similarly, this is a smaller device. You can see it has a concentric electrode. So inside this tube we have concentric electrode. One electrode is connected to a positive, and another electrode is connected to a negative end of a high voltage power supply. And in between this concentric pipe, you can see a plasma generation. So you can see we have placed a mirror at the bottom. You can see the plasma, which is plasma ring, which is generated as the end as we switch on this high voltage power supply. So this is a very small plasma device. And in a dark mode, you can see the plasma ring. So this plasma ring, it continuously generates plasma inside. The idea is inside a concentric tube, the plasma is continuously generated, and when you are connecting it to a air pump. So we are providing an air pump here. So it passes the gas from top to bottom. Means continuously the gas get ionized and it come back here. We can collect the ionized gas here. Okay. So this when the ionized gas is exposed to water, or when it is directly pumped towards the water, it make the water as a plasma activated water. It takes time like 1 hour, 2 hour to dissolve this reactive species into the water, and it make plasma activated water. So this is a small setup which make plasma activated water in a small laboratory scale. So we have connected this plasma. We have a concentric plasma electrode here. In between we have generate plasma, and when you pass the gas over from top to bottom, it collect the ionized gaseous molecules and it dissolves in the water. Thereby it produces plasma activated water. So you can see this is the setup where you can see plasma ring which is generated plasma which is generated in a concentric electrode. So if you see, so far we have discussed how to generate plasma, different techniques to generate plasma. Now application of plasma in food industry, as I said, the first is surface cleaning and decontamination. If you want to clean the surface, we use UV sterilization, right? When you are subjecting a UV light over a surface, UVC, which has a disinfectant effect, it destroys the surface microorganisms. Similarly, we can allow this plasma to sterilize the surface. We can allow the plasma to come in contact with the surface. It completely destroys the surface microorganisms and eradicate not only the microorganisms, even the chemical component. Pesticides, any molecules comes in contact with the cold plasma, it disintegrates the molecular structure. As the plasma is highly reactive, it immediately reacts with any materials and it degrades the component. Like as I said, it can be used to degrade toxins and the pesticide residues from the surface of my fruits and vegetables, and alter the rate of germination. As I said, it is used as a seed treatment, as it has a etching the surface of seed coat. It can be used as a seed treatment to germinate, to increase the rate of germination of seed. Modification of starch. It alters the structure. It alters the starch structure by the reactive species. When the reactive species are mostly the nitrogen reactive species, when it combined with the starch molecules or carbohydrate structure, it degrades the changes. It breaks down the structure and it modifies the structure. So manufacturing of resistant starch or modified starch. So in all those industries we have plasma as an application. Structural modification of food packaging materials is also having a application in case of structural modification. Inactivation of microorganisms and enzymes. When it is exposed to microorganism, it destroys the microorganisms, as well as this enzyme inactivation is also one of the major application in case of food industry. As I said, this this plasma treatment can destroy aspergillus species of fungus, and it also destroy the fungal toxins which is already produced, or the toxins, residues of the toxins present in the food materials, especially in case of onion, garlic, the black color fungus, or in case of groundnut and chili, where already the aspergillus species or aspergillus fungus have produced toxins. The fungal would have removed at any post-harvest treatment, either by cleaning or drying, alternate drying and cleaning. The fungal population or the fungus may be completely removed, whereas the residue or the fungal residue or the toxins which is produced by the fungal remains there. In such case, the cold plasma can be used to eradicate the fungal toxins which is produced in the food materials. This is an effective method of removing the residuals present in the food material. And as I said, cold plasma in microwave inactivation, you can see the surface after exposing the plasma for 2 minutes, it can brought you about 3 log cycle reduction in E. coli. This treatment prevents the growth of mycelium in the surface of fruits, or in case of bread, baked product, cut fruits and vegetables. So you can see salmonella, it can produce 3 log cycle reduction in case of salmonella and listeria, monocytogenes. So 3-minute or 2-minute exposure to plasma can bring you 3 to 4 log cycle reduction of microalgae population. The advantage is when you are exposing it for a 3 minute to a higher temperature, it degrades most of the color component, most of the other nutritional components are get destroyed. Whereas in case of cold plasma, it retains most of the nutritional parameters and it destroys the microorganisms. So impact of cold plasma in enzymatic activity. So there are a lot of studies which shows the effect of plasma in controlling the enzymatic activity. You can see when you expose the sample of cut apple, 10 minute, 20 minute, 30 minute, uh, there is approximately 65% reduction of browning area for 35 minutes as compared to control samples. So as you expose this cut surface to the plasma, it reduces the enzymatic activity in case of cut fruits and vegetables. Similarly, for other fruits also we can use the same plasma for controlling the enzymatic activity. The increase in the treatment time eventually decreases the polyphenol oxidase residues up to 42% in case of sweet potato and potato slices before dry frying. So this shows that plasma treatment of cut apple has low metabolic activity than control sample. So a similar effect can be observed in case of sweet potato and potato sauce as well. So in case of this um, industrial application, if you see when you before frying this potato chips, if it has this less polyphenol oxidase, we can produce clear white color chips. If you destroy the enzymes completely before going into the frying operations, it can avoid the brown spot where after frying it reduces the value of the chips. So that can also be one of the pre-operation can be used. And then the bacterial side effect of plasma in plasma activated water. We can see the surface changes. So this is a control sample. This is a you can see these microorganisms. The surface when it is exposed to plasma, it destroys the cell wall. It destroys the cell wall component. It allows the cell wall to ooze out, cell wall content to ooze out, and you can see that is the cell is completely destroyed when it is exposed to plasma. So this is plasma activated treated and this is control signal. You can see this very huge difference in the cell structure. And this is one of the study which we did in our lab, which enhancing the shelf life of green chili. So the green chili, the capsicum which we have taken is jalapeno and this pusa javala. This is a two variety of plasma which two variety of green chili taken to the study. We made plasma activated water like 20 minutes plasma activated, 40-minute plasma activated, 60-minute plasma activated. The 20, 30, 40 minute is nothing. We make plasma first and we allow the reactive species or the electron, singlet oxygen or other reactive species which is produced by the plasma to get dissolved in the water. So we continuously allow the plasma gas to come in contact with the water. Using a bubbler, we enhance the rate of dissolving of this reactive species into the water. Once it is activated for 20 minutes, 40 minutes and 60 minutes, we have taken the plasma activated water and dipped this green chili for few seconds and then removed it. Similarly, it is treated with cleaning solutions, commercially available cleaning solutions like pesticide removal solutions or vegetable wash, commercially available vegetable wash solutions. So, and then we have tested the quality parameters. Like for 60 days, we have stored these green chilies and evaluated the parameters. So you can see in case of 60 days, This is two different variety which is available in our market. The green chili which is treated for 60 minutes of plasma, 40 minutes of plasma, 20 minutes of plasma for 60 days. Even after 60 days, the freshness is retained in case of plasma activated water. 40 minutes, you can see the 40 minutes plasma activated water shows a better results in case of color retention. Whereas this is control sample and the cleaner solutions. So these are some of the cleaner solutions which is available in the market and the plasma activated water. And you can see again in case of another jalapeno variety of green chili, it also retains the freshness and color of the green chili. And if you see changes in the water, so when you expose this water plasma to the water, you can see the decrease in the pH and increase in the oxidation reduction potential of water, and the reactive species concentration. There is an steady increase in the reactive species concentration in the water, oxygen reaction species and then nitrogen reaction species. So we have made 20 minutes, 40 minutes and 60 minutes. So there is a steady increase in the reactive species concentration in the water, and we have dissolved oxygen and dissolved nitrogen species. So both you can see an increasing trend with respect to 20 minutes, 40 and 60 minutes of exposure to plasma. Similarly, the pH reduces and then the oxidation reduction potential decreases. And we have when we experiment with this green chili, you can see 20 minutes, 40 minutes and 60 minutes treated with some cleaning solutions and control. So at 21 days, the weight loss, you can see the weight loss. At 21 day, you can see a weight loss, a higher weight loss of almost 15% in case of control sample and the cleaner solutions. Whereas in case of plasma activated water, 20-minute plasma activated water and 40-minute, 60-minute, there is no much, less than 5%. Weight loss is less than 5%. Similarly, in another variety, also we have less than 5% of weight loss, and we have less microbial growth. When compared to control sample and the cleaner solutions, it doesn't have any antimicrobial properties, whereas the plasma activated water shows a very high microbial reduction in both the varieties. And if you see the quality parameters like firmness, spoilage percentage, the firmness of the chili, again the plasma treated shows less changes in the firmness when compared to the control samples. And the spoilage percentage, the spoilage was in control is about 35%. 35% is spoiled, whereas plasma activated water 20%, 40% shows less than 10% of spoilage, whereas for 20 and 60% is slightly higher, 12% of spoilage was seen in green chilies. So you can see how effectively this plasma activated water can be used to extend the shelf life of fresh produce. So similarly, any food materials, any fresh fruits and vegetables, like we have tried with strawberry, we have tried with lemon, it shows very good results. It can be effectively used to extend the shelf life of fresh fruits and vegetables, especially plasma activated water. Okay, as such plasma have different application, as I said, modification and then seed germination, modification of starch. We are not going in depth into those aspects, but as such in case of plasma activated water can be used for as a vegetable alternate to vegetable washing or cleansing solutions. This is one of the potential application of plasma activated water. And then we have plasma activated ice also. The concept of plasma activated ice is gaining more important nowadays. Instead of using normal potable water ice, if you make ice out of plasma activated water, it retains the reactive species as such for a longer period of time, and it can be used to enhance the quality of meat product. When it is ice packed, especially the seafood, when it is ice packed, it can retain the freshness of the seafood.
Good afternoon, sir. My question is, does this cold plasma treatment work well on all types of food? I mean, solid, liquid, semi-solid, like that.
It work like it's a surface operation. Mostly in case of solid food materials, when it is exposed to the surface, it makes changes, it modifies the, if it is used for modification, it is giving good modification in the surface. And again, when it is exposed to a liquid, it is used to like it is used in case of uh, extraction techniques. It degrades the cellular molecules and it degrades the microorganisms. Even the plant material, it degrades this, it break down the even the plant cells.
Okay.
So there are different type of applications. What I am talking here is, as we are talking about post harvest handling, one of the important application of this plasma is plasma activated water is it can be used to uh, extend the shelf life of fresh fruits and vegetables. One is, the first thing is it destroys the surface microorganisms, and it reduces or it removes the pesticidal residues from the surface. So these two major advantages are there when it is used as a plasma activated water and it is used instead of cleansing solution or pesticide removal solutions.
Okay. So one more question. As a somewhere in the slide, you have mentioned the mechanism of cold plasma on microbial deactivation. Okay. There is mentioned that the protein denaturation and one more thing, oxidation of amino acids. So there might be chances of happening such effect on in case of food also.
Food materials
And,
also.
when we apply it to the food rather than
That's what it is widely used for modification, structural modification of protein, starch. Not only the microorganisms, even the plant materials and the plant protein get denatured or altered in structure. The only thing is, the only way out is each and every reactive species or each and every, we have to characterize a plasma for a specific application. We have to produce a plasma which is having a reactive species of our interest which do whatever we require. Like if we want a modification, a structural modification, we need to make a characterize a plasma in such a way that it doesn't make changes in the other molecules, but it allows modification only. Such kind of plasma, more study has to be done in the plasma, characterization of plasma, and the particular plasma can be used for the particular application.
Thank you, sir.
If you are using a generalized plasma, it has lot of disadvantage.
Thank you.
Can plasma be used with other kinds of combination techniques like coating, plasma activated water as well as coating for preservation of fruits and vegetables? Is there a possibility for that?
Yeah, there are possibilities. Plasma itself a difficult task. It takes a like when it is comes to plasma, I am bringing this plasma activated water because if it is a plasma alone, generation of huge plasma is a difficult task first. Uniform exposure of fruits and vegetables. Like if you are exposing to a pin plate plasma, the one side of food materials will get exposed. One side of the fruit will get exposed. Other side of the fruit remains as such. So we need a mechanism to tilt the fruit through all the direction to expose to the uniformly to the plasma. Rather, we have to improve the design of plasma setup, or else we go for this plasma activated water. When you dip it in the plasma activated water, it covers the entire fruit. Like if you are talking about a larger fruit, we are talking about a larger fruit like apple or a pomegranate, we need an uniform exposure of plasma through entire site. That is the first difficult task we have. And then combining with other techniques can also be possible. Like initially we treat it with a coating solution, or then we could bring back to the plasma, or else first you treat it with a plasma and then you can take it to a coating solution, dip it in a coating solution. That is also that will also work good. Industrially you can combine.
The thing is, won't the reactive species interact with the coating solution and
No, no. It um, combining in the sense it has to be one after another, not at the same
same
time.
time?
Okay.
Okay, so, and I have a doubt on the slide.
Yes, please. Which
Um, what is EC, sir, in this? TDS and the other one is EC.
this is that is electronic oxygen reduction potential and the TDS, electrical conductivity of the solution.
Oh,
Okay.
okay, sir.
So when there is a changes in the electrical oxygen reduction potential, it alters the electrical conductivity of the solution. Okay.
Thank you, sir.
Thank you.
Good afternoon, sir.
Yes, please.
So my question is, you told us about plasma activated water. So we will treat water with cold plasma, right? So means for how long time it will take the properties of plasma? Like we will keep water in a normal way as we are keeping, or we have to keep it in a particular condition with precautions and all. And for how long time it will take the properties of plasma? And,
As per for 24 hours, we have seen there is no much changes. Once the oxygen species or nitrogen species get dissolved in the water, it remains in the dissolved state if it is closed, if it is completely closed and undisturbed
okay.
for 24 hours.
Okay.
If you are storing it under for a long time, definitely the reactive species will combine with other like it degrades.
Okay.
Um, Up to 24 hours there is no much changes we have seen. Maybe after 24 hours the dissolved oxygen species, the dissolved reactive species, the quantity of dissolved reactive species will decreases.
So
It will be in a decreasing trend
in a room temperature.
in a room
normal
temperature,
condition.
but up to 24 hours there is no much changes. If you see after 24 hours, it won't suddenly drop. The quantity of uh, dissolved species in the water will get degraded slowly.
Okay.
Maybe after 5 days or 6 days, you may not have any dissolved reactive species in the water. Okay.
And sir, you told about plasma activated ice. Then what is the concept behind it?
That is a new concept coming around. There is no much studies in plasma activated water. If you freeze the plasma activated water, that's the same question. It's connected with your previous question also. The degradation of reactive species, the rate of degradation of reactive species from the water can be reduced. The rate of degradation of reactive species from the water can be reduced when the liquid matrix is converted into a crystalline structure. That is plasma activated ice. Again, the reactive species can be preserved for a longer time. When the ice melt into water, the reactive species again it can be used wherever it is essential. That is what the ideology behind the plasma activated ice is. Like when you use this plasma activated ice for packing the fish, especially frozen prawn or fish, when the ice melt into water, it has the reactive species dissolved in it, so that particular reactive species can arrest some microbial growth, right? That is what an idea, but there is only less studies in this particular area. Even in our lab we are trying to make some plasma activated ice and study the property of plasma activated ice first. Still there are in other part of the globe there are people doing the research. um, still still the results available are very less. Nutritional losses. vitamins, um, there are there are nutritional losses. There may be nutritional losses. If it is, as I am telling about the largest fruits and surface treatment of fruits, right? Um, we are doing this plasma treatment along with the fruit peel. Like as I said, pomegranate. Pomegranate is treated as such with the water.
Yes.
So only the surface changes are there. The nutritional characters of the fruit inside is retained as such.
Yes.
If it is cut vegetables, for example, as I said, exposure of
Cut-
cut apples
cut
to
apples.
avoid enzymatic degradation.
Yes.
In that case, again the answer is the same as previous question. We have to select a plasma type of plasma in such a way that it do whatever we require alone. The reactive species, or either we have to select, we have to carefully choose the gaseous composition which we are taking for a particular operation. Right?
Yes.
Either we go for selective production of reactive species in plasma production, and then do our job. So it alone, it destroys the enzymes alone, and it doesn't react with the vitamins.
Yes.
In that way, we have to make a plasma in such a way that it doesn't destroy the enzyme, it doesn't destroy the vitamin component, whereas it only destroys the enzymatic components. Okay.
Thank you, sir.
Thank you.
Good afternoon, sir. My question is, at what temperature range does this cold plasma process carried out?
It is atmospheric temperature only. If you generate plasma, if you expose it to a surface for
Yes, sir.
3 minutes, 5 minutes, the temperature may increase from, if it is initial temperature is 30, it may go to 32, not more than that.
Okay, sir.
Like if we are continuously exposing to plasma, a surface temperature can go, maybe increase to 2 to 3 degrees Celsius. That's it.
Okay, sir. Thank you, sir.
Thank you.
Thank you, sir, for your valuable time in explaining and providing inputs on application of cold plasma, plasma activated water for preservation of fresh produce. Herewith, we are concluding our today's theory session with introduction of plasma, technical characteristics and influencing parameters in plasma treatment and effect of plasma activated water on quality parameters and properties of food components. Sir, provided insights through the video about the working mechanism of dielectric barrier discharge, pin to plate plasma device and its application in food industry with case studies. Once again, thank you, sir.